In this section, I want to introduce concepts on how scientists, medical professionals, clinical laboratory professionals diagnose infections in a laboratory setting. As we continue to talk more about very particular types of infections, we will talk about how those are more specifically diagnosed. Very commonly, clinical laboratory professionals will need to use microscopic methods in order to diagnose infections. And there are a number of types of microscopy that can be used, and we'll go through those in the next few slides. Light microscopy is the most commonly used form of microscopy. This is what you've done in all of your classes, that standard little microscope. When we use bright field microscopy, we use visible light to basically illuminate the background, and then the cells are seen as darker on our field of view. So the background is lighter, the cells are darker. This type of microscopy has the lowest resolution because of the light wavelengths. It has the longest light wavelengths. So this is the physics of microscopy that you should have already learned in a number of different classes. So you should understand these concepts of resolution and the wavelengths of light. Dark field microscopy is very similar to bright field in terms of it relies on light in the visible spectrum. However, instead, what we do is we make the background dark and light up the samples. So you can see this is basically the exact same image under light or bright field microscopy and under dark field microscopy. We will sometimes rely on dark field microscopy because it does provide increased resolution. You can see the outer structures of these cells a little bit more clearly, and it's commonly used for visualizing spirochetes, so Treponema, Leptospira, and Borrelia, because those microbes are so very thin, it can be hard to see them using bright field, but they can be visualized using dark field microscopy. We can also use phase contrast microscopy. This is good if your organism is still living, if you've done something like a wet mount. This allows for imaging of internal structures and can give a 3D visual of live samples. And again, it takes advantage of the different refractive indices of various parts of the cell to allow that type of visualization. Fluorescent microscopy is also very common, but we begin to get more expensive as we move from regular light microscopy to phase contrast to fluorescence, and then what we'll talk about is electron microscopy next. With fluorescence microscopy, you use what are called fluorophores, or compounds that will emit and have different wavelengths of light, basically, so you can see them as different colors. So we use these fluorophores to tag various structures. One of the most commonly used is DAPI, which eventually will stain the nucleus, but you can have fluorescently tagged antibodies that will bind to various antigens on a cell surface or even internal into the cell. This is an image that I took using confocal microscopy. This is a live dead stain. Live bacteria are green, red bacteria, the dead bacteria are red, and these are actually Borrelia burgdorferi, the organism that causes Lyme disease. So you use one wavelength of light to excite the fluorophore, and then as that excited fluorophore kind of comes down from that excitement, it emits a different wavelength of light, and that's the color that you visualize. Electron microscopy allows for really high resolution. Electron microscopy is the only way that you're really going to be able to visualize viral samples. There are two types of electron microscopy. Again, you should have learned about these already. There's scanning electron microscopy and transmission electron microscopy. Transmission electron microscopy, or TEM, has the highest resolution compared to scanning electron microscopy, but they're both very good. And so as you see in this top image, this is Staphylococcus. This is actually a biofilm of Staphylococcus aureus that I grew. I didn't do the imaging, but I did grow it up. And so you can see, the details of some of the connective structures that they have with one another. components of the biofilm. You can see some that are in the process of dividing. And then this is a transmission electron micrograph where you can see even more detail of the surfaces. So this relies not on visible light, but as it says, electrons. It's a beam of electrons. And because those have such a short wavelength, you get really significant resolution. When you are using microscopy, something to think about is larger organisms are much easier to see. If you're trying to diagnose somebody who has a fungal or parasitic infection, microscopy can be fairly straightforward because those organisms are usually large enough to see without any sort of preparation. For smaller samples like bacteria or even some of the small yeast, you may need to do some preparation of the sample, so it requires help for you to be able to see those. And some of that help that you may have to do is different types of staining. So here it talks about how you can just look at wet mount, direct examination, but you should remember from Bio 120 that there are various types of stains. There's simple stain, negative stain, of course our selective and differential stain, like a gram stain to determine whether or not the cell has one membrane or two, or a thick layer of peptidoglycan or thin. There's the Zeil-Nielsen stain, which is also sometimes called the acid-fast stain for visualizing cells that have a lot of mycolic acids in the cell wall. And then there's an endospore stain that allows the spore to be stained green and the vegetative structure to be stained pink. There's also fluorescent staining that you can use, as we saw in a previous slide. Another trick or tool that clinical laboratory professionals will take advantage of is culture. Sometimes culture is super easy, especially with some of the more common bacterial infections, E. coli, salmonella. I mean, those will grow on a plate, no problem. It can be more difficult to culture other organisms. Some organisms require you to use regular cell culture, like tissue culture. For example, intracellular bacteria, they won't grow on a plate. You have to grow them in tissue culture with eukaryotic cells. Viruses also require cell culture. You can't just grow them on a plate. It can be much more difficult to isolate those types of organisms because you do have to grow them in cell culture. And there are other microorganisms that are maybe a little bit more particular in the type of growth media that they want to use to grow. So they can be more challenging to culture as well. So some are going to be really easy. They'll grow on pretty much any kind of plate. Some are going to be more fastidious. You have to have specialized plates in order to grow them. And some won't grow on a plate at all. You'll have to grow them in cell culture. Various types of culture media are listed here. There's non-selective, which really should grow pretty much anything, like blood. Will grow a lot of the real common bacteria and fungi. We start to get more specialized. Chocolate agar is a type of, it's not chocolate. I know it sounds good. You're like, chocolate agar. No, it's cooked blood. So it's the color of chocolate, but it is not at all chocolate. And sometimes you need that type of media to grow a little bit more fastidious organisms like Haemophilus or Neisseria gonorrhoeae, because they're not able to break down the red blood cells. You have to heat it up to break down the red blood cells. There's Mueller-Hinton that you would use to do bacterial antibiotic resistance assays, thioglycolate for culturing anaerobes, etc. We also have selective and differential media. For the most part, most of our selective medias, especially the ones that you would have used in Bio 120, will be selective for gram-negative. So McConkey, EMB, they'll only grow gram-negative. And then they're differential because the various gram-negatives will look different. But another one that you might have used in Bio 120 is mannitol salt agar, or MSA. So that is selective for Staphylococcus. It only grows members of Staphylococcus. However, it's differential because Staph aureus should turn yellow. So you have those selective and differential media, and then down towards the bottom, You see we have specialized media. So we have BCYE agar that will recover Legionella and Nocardia. You have specialized agar to grow Corneobacterium, diphtheriae, specialized broth for growing Strep A. galactiae, Bordetella pertussis, Vibrio. So we can get very specialized in our type of media. So you, as a medical professional, clinical laboratory scientist, need to have some idea of what type of infection your patient might have so that you can select the appropriate type of media. You're not going to be able to culture a patient's specimen on every type of plate. So it's really important that you are able to think about the type of infection your patient might have and how that can lead to different types of growth media being necessary.